All right, hello everybody. Thank you everyone for joining our webinar. My name is Ohimai Unoje, and it is my pleasure to present to you a little bit about some of the exciting things that we're doing in the space of cell preparation for flow cytometry. The title of my talk today would be Next Generation Cell Preparation with Laminar Wash Technology. And what I mean by next generation cell preparation is the idea that when we think of our sample prep, the opportunity to prep our samples a little better. You know, if you think of what next generation sequencing has done with traditional sequencing and greatly improved what we're able to do with it, the goal is to do the same thing with our sample prep. And in this webinar, my goal today, number one, is threefold. We're going to discuss the challenge and the status of manual sample prep. And then we're going to look at how laminar flow can benefit flow cytometry sample prep. And then lastly, what I'd like to do is go over some data highlighting some of the impacts that laminar wash has had in flow cytometry. So let's get started here. If you think of the work that are being done in your scientific labs, you know, you guys are doing incredible work with looking at analytical tools like flow cytometry, single cell sequencing, to do important things in a variety of fields: immunotherapy, cell and gene therapy, vaccine analysis, you name it. Labs are taking very heterogeneous, oftentimes complex biological samples, having to go through a sample prep, which currently is a very manual process, can often be difficult to automate, often introduces user variability. Sometimes they can be even harsh on cells. And so what we're proposing here is the idea that what if that sample prep process could be automated, could be made to perform with as minimal user interaction as possible, make it more accurate, reproducible, fast, while still being gentle on the cells. Now, if you have that kind of sample prep, now then imagine what that could do to your cellular analysis and impact downstream what it is that is being done. And so that is where the laminar wash technology comes in. It is a unique, innovative technology that leverages a very unique plate design of hydrophilic surfaces in the well, surrounded by a hydrophobic surface, which enables this wall-less well. Each well can accommodate up to 80 microliters and up to 10 million cells in each well. So the users would load their samples into each well, and then after the samples are loaded in the wells, the cells in there begin to settle gently by gravity. There are no physical forces, no extra physical forces in place. This also means that when you're doing your cytometry staining, your antibody interaction with the cells is a natural interaction, not influenced by external forces, but it also minimizes the interaction and the settling of unbound reagents or cell debris. So you get the separation between your stained cell of interest and your unused reagents and debris. After the settling process, the plate is then loaded into the system for washing. And the user can then set and choose their flow rate, the number of washes, pretty much the wash process, completely customizable according to the user. And what happens is the system will introduce two nozzles. One nozzle introduces new buffer, and then another nozzle aspirates out the buffer. And as you continue to repeat this process, what you're doing is you're creating a laminar flow that actively diffuses the debris that is unbound to aspirate it away while keeping your cells of interest retained at the bottom of the well. So you have this gentle wash process that does not disturb your cell, but efficiently and effectively cleans out your debris. So when you think in flow cytometry, one of the things we know in flow cytometry is that each protocol for sample prep is different. But the good thing with the laminar wash is that it is easily customizable, or different protocols can be adapted to laminar wash. This is a protocol that we received from a customer of ours, showing on the left their current sample prep process, and then on the right what that time process looked like with laminar wash. And the total time for their protocol that they spent washing per plate was 42 minutes with their manual process, compared to about 17.5 minutes with the laminar wash. This is not the only protocol. We've received other protocols as well, here showing that this other group were able to go from reduce their sample prep protocol from taking about 60 minutes to about 6 minutes. So one of the exciting things that we've seen with the laminar wash is it enables users to really cut down their sample prep time, meaning they're getting to their analysis and their runs faster. But not only that, the laminar wash plates can be directly analyzed on most cytometers with auto samplers and plate loaders. So oftentimes there is no need for, oftentimes we can avoid that process of transferring from plates to tubes before loading. And what users have observed is they have seen improved staining, resolution, population separation, and better MFIs. So all of this is just to say that the laminar wash system has really done a lot of good within the field of flow cytometry. However, I should say, moreover, we've seen application of the system beyond flow cytometry. And here are just a few of some of the life science disciplines where laminar wash has been applied and individuals are seeing the benefits. Single cell sequencing, immunoassays, mass cytometry. However, one of the things I like to share about the laminar wash, especially about this application, is if you do not see your technology or your scientific discipline here, please reach out to us. Contact us at Curiox, because there's a good chance if you're working with some cell-based analysis, protocol, process, then there is a good chance that the laminar wash system could be adaptable to the work that you are doing. So this is a brief overview of the laminar wash system. Next, I'd like to go in and show a few cases and data showing how laminar wash has improved some of the research that people are doing all over the country and all over the globe. So this first case that I'm showing here is a group that wanted to better characterize the immune cell population that infiltrate a tumor after the introduction of therapy. So in this case, they introduced a tumor cell line into a mouse, had that generate a tumor, introduce a PD-1 antibody or a vehicle control with the goal of then analyzing the immune cells that came in, that infiltrated the tumor. So the tumor was excised from the mouse, dissociated into single cells, and then the staining and wash and run were all performed on the laminar wash plate. And then went in for flow cytometry analysis. And right off the bat, one of the most striking observations they observed was that, and if you look in that top right panel, increased viability in the number of cells that they were working with compared to the manual or the traditional way of sample prep. But that was not the only benefit that they received, because if you then look on that bottom panel and looking at their flow data, one of the things that they observed looking on that side scatter, forward scatter gate, that first two plots, they noticed that the amount of debris that they recovered was much lower than what was than what they got with the centrifuge. So this told them that with the laminar wash process, they were able to get better debris removal from their samples. But debris removal was not the only benefit they observed, because they saw a significant increase in the amount of viable TILs. Now, for anybody that's working with either tumor-infiltrating lymphocytes or macrophages or immune cells that have infiltrated the tumor, we know that the isolation of these cells can be challenging, especially of viable cells. And so they were very excited to see an increase from about 60% viability to about 80% viability. The other thing that they also pointed out was that looking at the percentage of CD45 expressing immune cells, we did not disrupt that ratio, letting us know that even though as we're increasing the amount of viability, we are doing this without introducing a bias into their data, without compromising the expected data. We're giving them good data, expected data, but just making it better. So when we reached out to the lead scientist who did this experiment, this was the feedback that he provided. They observed improved workflow, reproducibility, consistency, retention, viability, and even better resolution of their cell populations. Lower stress on their sample, lower debris and autofluorescence, lower clumping, lower single cell suspension volumes. And for those anybody here working with animal models, we know we are motivated to perform science on our animal models with the most ethical treatment of those animals. And that is what the 3 R's is. And by doing this, we enable them to reduce the amount of animal waste or unnecessary usage of animal sample, which is that 3R commitment. So that was an exciting result for them. Next, I want to go to another data from a research lab in Maryland. And this group wanted to look at lymphocyte retention, but rather than from a tumor model, they wanted to look at it from bone marrow and spleen samples. And immediately they observed, again, increased amount of cells from the laminar wash process compared to the centrifuge, meaning the laminar wash system enabled them to retain more of their samples. And we know in this field that we're working, I like to tell people, each cell lost is data lost. So to be able to gain about tenfold, about 50% more of the data that they were getting, they were very excited for that. But not only were they excited about the increased cells that they were getting, when they looked at the actual populations, they saw increase in each and every one of those immune cell types without compromising the population ratio. So once again, we're improving this. We've shown it in tumor samples. We're also seeing it in bone marrow, spleen samples that increase viability without compromising the actual data or without introducing a bias. Next data I'd like to show you was a group that wanted to improve their user consistency. So this is a group that does that generates cell therapy, cells for cell therapy. And they had multiple users in their lab, and they wanted to ensure that regardless of which user is performing the cell recovery process, the results were reproducible and repeatable. So using our laminar wash, they had an operator with expert level proficiency and a new user in the lab perform the same experiments recovering the different type of cells. And what was observed was that across the board, by using the laminar wash, we could eliminate that skill difference, that retention that was based on difference in skills. So whether an experienced user or a new user, they were able to recover similar and repeatable percentage of cells. What was also remarkable for them was in this population here, this gamma delta cells, these are very rare population that were less than 0.5% of the cells, difficult to recover, often difficult to be reproducible. And they noticed that the laminar wash system introduced a level of consistency in there. And as you can see, their director of development commented that it allowed a consistent analysis of cells that are less than 1% of the population over and over. Now, this other piece of flow data that I'd like to show, similar to everything we've talked about, is the idea of better recovery. However, these were with cryopreserved samples. And the cryopreservation and thawing process, we know, can be fairly harsh on primary samples. In this case, we observed similar impact. We showed an increase in viability from about 68% with the traditional process to about 80% with the laminar wash. But also certain populations of interest to this group were increased. We see increase in their amount of monocytic cells, dendritic cells, and even their natural killer cells. So we're able to improve the viability from difficult to handle cryopreserved PBMCs. Now, this last piece of data that I'm sharing, we're actually switching from flow cytometry to single cell sequencing, because oftentimes when users go through a flow-based sorting, there's a lot of interest in understanding what's going on, the biological phenomenon at the single cell level. And so this was a group that wanted to improve their single cell recovery. And what they observed was, using the laminar wash system compared to the centrifugation, they saw almost a doubling of the amount of cells that were recovering. And if anybody here is familiar with the concept of total-seq, it's the idea that you can perform multi-omics and look at multiple omics data from the single cell. A key aspect of that is the ability to recover true single cells. Well, what was observed was that we minimized the amount of doublets while increasing the amount of singlet population in their sample. We also reduced the number of negative population in there. So what that means is, when they're doing their single cell sample, they have less waste in their single cell analysis and more actual data to work with. Now, this is something that as a part of the Curiox team, I am personally proud of. BioLegend, who manufacture and own the total-seq technology that is used for site-seq and other single-cell multi-omics, implemented our laminar wash system as part of their protocol. And their conclusions were, they saw an improvement in the speed of the sample prep compared to centrifugation. They also observed that the data either looks identical or better. So there was no loss in data quality from switching to Laminar Wash. They were even able to identify the ideal parameters for their particular workflow. And now, as of December 2020, proud to say that the Laminar Wash system has now been incorporated as part of the BioLegend Total Sig official workflow, just because of the benefit that it brings to that workflow. So to summarize, Laminar Wash technology is the next generation in sample prep, and the improvements that it's bringing and it's going to continue to bring range from workflow improvements such as the time savings, consistency, even biosafety, because this system can fit one into a biosafety cabinet, and because there is no need for plate flicking, there is therefore no aerosolization and lower exposure of the user to any biosafety samples or biohazardous samples. But at the same time, we're also seeing better quality in data because the cells are in their native state. We are increasing the amount of cells that users are getting. We're removing the amount of debris, so there is more actual data and less waste. And because it's a better cell washing, we see a higher signal to noise ratio. We see better antibody binding. We see enhanced staining efficiency. So this is what Laminar Wash brings to the scientific community. So my next few slides are going to go a little bit over what the Laminar Wash system looks like. So here at Curiox, we have three main systems. We have our mini system, our HT2000, and then our Auto 1000. Our HT2000 is the flagship system that we have. It has the capability of washing 96 well at a time, 96 well plates at a time, and can wash 12 plates in as fast as 26 minutes. I'd like to see a user try that and beat that. We see improved workflow with less hands-on time, meaning that there is less variation. You can have that multiple steps that occur with a centrifuge all combined into one. You get improved reproducibility, consistency, no aerosolization. It's a safer process, has a touchscreen interface. It also has an optional buffer exchanger for those whose protocols work with multiple samples. Next is the Auto, and really what this is, it's a fully automated sample preparation system for flow cytometry that introduces automation into a flow cytometry workflow that does not require automation experience. We at Curiox have designed a proprietary graphical user interface, which we call the GUI, that is designed for flow cytometry users who have no automation experience. If a user can identify their volumes and their reagents that they need and input that at the push of a button, they can use the Auto. That also means there's less manual sample handling. That means then you have more time for data analysis because the system would take care of the sample prep. It is compatible for both surface and intracellular staining, and it is fully automated with electronic reports for tracking. Oh, and did I mention there is no automation setup needed? It is off the shelf, ready to be used. For lack of a better phrase, it is the automation version of a plug and play. And then the last instrument that we have is the Laminar Wash Mini. And this was geared towards those that are doing single cell sequencing. Washes 8 wells at a time. It comes with, rather than a 96 well plate, comes with a mini strip of 16 of 2 strips of 8. Very similar input or very similar structure as the typical single cell platform for like a 10x genomics, but yet you get all the benefits of the Laminar Wash. So I want to conclude with this statement. Laminar Wash makes the user's life better. Better sample handling, better process, lower debris, better samples at the end of the day. And feel free to take about 20 seconds to read how some of our users have commented on how we've made their life, their lab life, just better. So if you would like to see this system in action, we have a system here with us at the University of Chicago Cytometry Core with David Leclerc and Michael Olson. If you're at University of Chicago, we would love to invite you to come join us. Megan, I believe we have a poll just for this. So users, Megan is going to send out a poll. If you would like to be a part of a demonstration today, please go ahead and indicate that on the poll. Let us know yes or no. I have about 5 more seconds left. Okay, Megan, let's go ahead and close the poll. So if you'd like to be a part of this, in the chat, Megan is going to post a link to this webinar registration page. At the bottom of that page, we have a demo registration form. Please go ahead and fill that form, and I will personally reach out to you after this webinar, and we will coordinate with you about how we can get you involved in the demonstration. We will be having our demonstration today at 12 p.m. at the Flow Core at the BSLC building room R022. So without any further ado, first of all, I want to say thank you to everyone who has listened to me drone on for a little bit. Now I am going to invite my colleague and our application scientist, Mark, and we're going to be taking any questions that have come in. So Megan, over to you.
Thank you, Ohi. That was a wonderful presentation. And welcome, Mark. It's wonderful to have you on. So while we're waiting for some questions to come in, I do have a couple frequently asked questions that we could start it off with to make things just run a little bit smoother. So, Mark, could you tell me how many samples can I run at a time? Does it have to be 96, or is that something that is flexible?
It's absolutely flexible. And thanks for having me here, everybody. It's a pleasure to be speaking and meeting you all, even virtually. We're excited to be presenting all this to you. With the laminar wash plate, you can absolutely reuse that plate for later studies. Of course, that doesn't mean you would want to reuse a well that you've already put sample into, because you wouldn't want to contaminate that with something that's already been run. But all of those unused wells on that plate are absolutely ready to be used for a later experiment.
Awesome. And I know we've gotten some questions in the past about this. Are the plates reusable, or are they a one-time use?
They are absolutely reusable. And so you can go ahead and wash off any residual buffer. Every well will be washed throughout the washing procedure. And so that allows you to store the plate and then come back to it within about a month's time and finish off the rest of that plate.
Awesome. So some people may be a little bit interested in how exactly the plate works. How do the wells not spill over to the next well without walls? And to go with that, are we able to vortex the plates?
So we do have a proprietary barrier that sits in between the wells. So the wells themselves are hydrophilic, while the barrier around the wells is hydrophobic. It's really quite remarkable seeing the droplets on these plates, because you can put a droplet size of up to 80 microliters. With the additional adapters that we have, you can increase that volume up to about 150 microliters. These plates are vortexable. We do produce a shaker as well. It has a very short throw, and so you can watch those droplets dance around and mix, resuspend your sample, whatever you need to vortex that sample for.
Awesome. So we got our first question in, actually. So have you tested the technology with unusual samples such as whole worm, planaria homogenerates? I'm sorry if I mispronounced that, but thank you, Kevin, for your question.
So, as far as I know, we have not worked with whole worm planaria homogenates, but that does not mean that they won't work. It's absolutely within the realm of possibility that these cells would be very capable of being washed with laminar wash.
And sorry, Megan, I'm going to add a little bit to that. So, Kevin, that's an excellent question. And that is something that I definitely would like to follow up with you a little bit more about, because as Mark has mentioned, while we may not have worked with that, we would love to see how the laminar wash system will work or can work with something like that. So if you don't mind, Kevin, I will be following up with you after the call to maybe we can have a sit-down conversation about your whole worm planaria homogenate and how it can work with the laminar wash.
Yes, thank you again, Kevin, for your question. So up next is a question from Bartholomew. Have you performed washes with fixed cells?
Yes, we have.
All right.
To add to that, there's so many things that you can do on the laminar wash plate, and we have mentioned as well that you can perform intracellular staining. So that does mean you can also fix your cells on the plate partway through a protocol without ever having to remove those cells or transfer back and forth between a plate and another vessel. That goes as far as being able to a direct read off of the plates at the end of a workflow by many flow cytometers.
Awesome, awesome. Thank you so much for that, Mark. That's an awesome addition to that. So while we're waiting for a couple more questions to come in, I can go ahead and give a couple more of our frequently asked questions. So, do our systems work well with nuclei?
Yes, absolutely. So for those people working on the single cell space, you can absolutely isolate nuclei and put them on the plate, or you can lyse them directly on the plate and proceed through any workflows like a 10x genomics chromium assay or any of the variety of others that are similar out there.
Wonderful, wonderful. All right, so we still have a little bit of time left, so if anyone has any other questions, do feel free to add them to the chat. We have some time remaining and we'd love to answer any questions that you have. Feel free to put whatever you want to ask.
I can say on the nuclei side, as well as we're waiting, that one of the major benefits that we get with laminar wash is less clumping. Of course, with nuclei and isolating nuclei, one of the things that we want to make sure of is that when we do process the cells or our samples, they aren't clumped, that they are truly single cells that we're looking at. And so by using laminar wash and making sure that we can keep those samples separated, that we are confident that the data we're getting is very nice, stringent, and something that we can take all the way to the end.
Awesome. All right, well, it seems like, oh, we got another question from Kevin, actually. Are the Oh, it looks like you're asking if the plates are reusable. I think we mentioned that just a second ago, but Mark, could you just give like a quick response on that again?
Absolutely. So you can absolutely reuse chips and plates. We don't necessarily recommend using wells that you've already put sample into, but any unused wells are something that you can come back to and use at a later date.
All right. Well, thank you everybody for attending today. It looks like there are no more questions. With that, I'm going to say thank you all for attending. Ohi will be there for how many days again, Ohi?
Yes, I will be at University of Chicago today, and we'll have the option of also being here tomorrow for anybody who's unavailable to speak with us today.
Wonderful. So thank you all again for showing up and enjoying this awesome presentation. We'll see you all hopefully next time.